Hi friends, welcome to biologyexamsforyou.com. Presently we are fighting against COVID-19 together. Please stay at home, stay safe and save lives. If you have any doubts regarding COVID-19, you can visit the WHO's website. The link is given in the description. May Almighty protect us all from this deadly virus. The topic of our discussion is the difference between Krebs cycle and Calvin cycle. Difference number 1 regarding the occurrence and site of reaction. Krebs cycle, as you all know, it is a stage in cellular respiration. Therefore, it takes place in all aerobic organisms. And the organelle involved is mitochondrion. So let us zoom in this mitochondria, and the exact site of reaction is the mitochondrial matrix. Whereas Calvin cycle is a stage in photosynthesis. It takes place in chlorophyllated cells or green plants. The organelle involved in reaction is the chloroplast. And the exact site of reaction is the stroma of chloroplast. Difference number 2, overall reaction. In Krebs cycle, it involves series of reaction that oxidizes glucose producing carbon dioxide, ATP, NADH and FADH2. So after glycolysis, which is the first step of cellular respiration, acetyl coenzyme A enters citric acid cycle. In this reaction, glucose is oxidized, therefore it is a catabolic reaction. So this is the reaction cycle. As you can see, there is NADH is produced, and then there is FADH2, PGTP or ATP, then there is FADH2. Krebs cycle is considered as an aerobic process as the substrate for this cycle, that is NAD plus and FAD, is recycled only in electron transport chain where oxygen is essential. Therefore, Krebs cycle is considered as an aerobic process, even though oxygen is not directly involved in the process. Next is Calvin cycle. In Calvin cycle, glucose is synthesized where ATP and NADPH are utilized. As you can see, this is a light reaction that occurs in the grana where ATP and NADPH is synthesized, and sunlight, the energy from the sunlight is converted to chemical energy, and that energy, ATP and NADPH, is used to convert carbon dioxide to glucose in Calvin cycle. Therefore, it is an anabolic process or glucose synthesizing process. As you can see here, ATP and NADPH are utilized, and finally glucose is synthesized in the process. And it is an anaerobic process also, doesn't require oxygen. Difference number 3, major steps and enzymes involved. In the case of Krebs cycle, the first stable compound is citric acid, therefore also called a citric acid cycle. As you can see, oxaloacetate, which is a 4-carbon compound, combines with acetyl coenzyme A, and the enzyme involved is citrate synthase, forming the first stable compound, citric acid, which is a 6-carbon compound, therefore also called as citric acid cycle. Enzyme involved is citrate synthase. During the process, ATP, NADH, carbon dioxide and FADH2 are produced. Whereas in the case of Calvin cycle, here RUBP combines with carbon dioxide, which is the first step in the presence of enzyme rubisco, ribulose biphosphate carboxylase oxygenase, one of the most abundant enzymes, and converts it into a 3-carbon compound, which is 3-phosphoglyceric acid. The first stable compound is 3-phosphoglyceric acid. Therefore, it is also called a C3 cycle. Enzyme involved is rubisco. Here ATP and carbon dioxide are utilized. Approximately 12 plus 6, 18 ATP and 12 NADPH are required to synthesize one glucose molecule. And that's it. Thank you so much for your support. You are with biologyexamsforyou.com. Thanks for watching.